discuss questions in the last aspect of cell biology, that is cell division completely. Cell division completely. Now, in anaphase of mitosis, whenever anaphase takes place, by the contraction of spindle fibers, there is a pressure created on centromere. Whenever a pressure created on centromere, every chromosome divides at the point of centromere. If every chromosome divides at the point of centromere, the approximate or diagram of anaphase is every chromosome divides into special chromosomes with one centromere, one chromatid and two arms like this. These are called daughter chromosomes. And these daughter chromosomes were pulled to their respective poles by the pulling action of spindle fibers. So, according to this question, the anaphase of mitosis, two strands of DNA get separated is wrong. Two genomes get segregated is wrong. Two chromosomes of bivalent get separated wrong, because in the case of mitosis, no chance of formation of bivalents. Two chromatids of a chromosome get separated is correct. Two chromatids of a chromosome means till metaphase, chromosomes show two chromatids and four arms. But from anaphase, chromosomes show one centromere, one chromatid, two arms. So, chromatids got separated. So, definitely they are called daughter chromatids or daughter chromosomes. Daughter chromosomes are pulled to their respective poles by pulling action of spindle fibers. This is a question regarding matching that to what you called meiosis. Repulsion between homologous chromosomes, diplotin, because in this stage the dissolving of synaptinimal complex enzyme occurs. Attraction between homologous chromosomes. Attraction between homologous chromosomes is called synapsis, which occurs in zygotin. So, answer is 3, A to B4. A is repulsion between homologous chromosomes, diplotin, because here synaptinimal complex enzyme starts dissolving. Attraction between homologous chromosomes is synapsis, where synaptinimal complex enzyme forms, that is zygotin. Exchange between non-sister chromatids. Any time exchange between non-sister chromatids occurs in patchitin. This exchange of chromatid bits is helped by endonuclease and ligase enzyme. Endonuclease enzyme cuts the part. Ligase enzyme, after the exchange, reattaches the bits, and this is patchitin, where crossing over takes place. Exchange between non-sister chromatids is also called crossing over, which occurs in patchitin. Separation of homologous chromosomes occurs. Separation of homologous chromosomes occur in anaphase 1, which is popularly called segregation or disjunction. Here the diploid set of chromosome become haploid set or genome, and homologous maternal and homologous paternal chromosomes get separate. So, repulsion between homologous chromosomes is diplotin. Attraction between homologous chromosomes is zygotin. Then exchange between non-sister chromatids is patchitin, and separation of homologous chromosomes is anaphase 1. There is one called diakinesis here. Diakinesis is known for terminalization. Terminalization. So, answer for this second question is 3. Assertion. Division of chromosome takes place in meiosis. Disjunction takes place during anaphase 1. Division of chromosome takes place in meiosis is correct, because in anaphase 2, every chromosome divides at the point of centromere to form daughter chromosomes. So, division of chromosome takes place in meiosis is correct, because it occurs in anaphase 2. Disjunction takes place during anaphase 1. Division of chromosome takes place in meiosis, disjunction takes place during anaphase, both are same, but because disjunction takes place in the anaphase 1, division of chromosome takes place in meiosis is wrong. So, these two are correct regarding meiosis, but they are not linked with each other. That is why answer is A and R are 2. R is not the correct explanation of A. Chiasmata are formed due to. Chiasmata are always formed due to, in the patchitin stage of meiosis, nucleolus, nuclear membrane still appear. Every chromatid arm of homologous chromosome splits longitudinally like this. So, every homologous chromosome gets one centromere, 2 chromatids, 4 arms. Every bivalent gets 2 centromeres, 4 chromatids, 8 arms. 2 centromeres, 4 chromatids, 8 arms. These 2 chromatids of this chromosome are called sister chromatids. These 2 chromatids of this chromosome are called sister chromatid. The sister chromatid of this chromosome become non-sister chromatid to the sister chromatid of this chromosome. I will repeat it. The sister chromatid of this chromosome becomes non-sister chromatid to the sister chromatid of this chromosome. Pairing occurs between non-sister chromatids, and at the point of pairing an X-shaped connection is formed called chiasmata. So here answer is overlapping of non-sister chromatids of between non-homologous chromosomes is wrong. Duplication of homologous and non-homologous chromosomes is wrong. Loss of some part of chromosomes is wrong. Overlapping of non-sister chromatid between homologous chromosomes is correct. So overlapping of non-sister chromatids between homologous chromosomes is correct regarding Pachitin, and chiasma are formed by the overlapping of non-sister chromatids between homologous chromosomes. Now, another matching. List 1 is zygotin, Pachitin, diplotin, diakinesis. This is also regarding meiosis. Meiosis is very important for MSET. List 2, movement of chiasmata, pairing of homologous chromosomes, formation of chiasmata, beaded chromosomes, repulsive forces between homologous begins. A2, zygotin, pairing of homologous chromosomes, synapsis. Pachitin, B3, formation of chiasmata. Formation of chiasmata occurs in Pachitin. Diplotin is 5, repulsion force between homologous chromosomes because the synaptinimal complex enzymes dissolves in diplotin. And diakinesis is movement of chiasmata. Whenever chromosome starts moving wide apart from each other, the chiasma connections move upwards and almost present at the terminal ends. This movement of chiasmata to the terminal ends is called terminalization. So answer is 4, A2, B3, C5, D1. Again regarding mitosis and meiosis. Anaphase 1, metaphase 1. Formation of 3 spindle apparati, formation of 2 spindle apparati, splitting of centromere, formation of 1 spindle apparatus, disjunction of homologous chromosomes. For this A is 3. Anaphase, splitting of centromere. Whenever splitting of centromere takes place, then only daughter chromosomes are formed. Anaphase 1, 5. Disjunction of homologous chromosomes. Homologous chromosomes diploid get separated into haploid set or genome, and genomes are pulled towards their respective poles. This pulling of genomes towards their respective poles with the help of spindle fibers is called as disjunction of homologous chromosomes. Then C4, metaphase, formation of 1 spindle apparatus. In one cell, only 1 spindle apparatus is formed during metaphase 1. Metaphase 2, formation of 2 spindle apparati. Why 2 spindle apparati? Because in metaphase 2, in each half of the cell, chromosomes get regularly arranged at the center on a metaphasic plate and spindle fibers are clearly visible. So 2 times spindle fibers are formed. So here the spindle fibers are formed exactly perpendicular to the places where spindle fibers are formed in metaphase 1. So the places to the metaphase 1, they are present exactly perpendicular to the places where spindle fibers are formed in metaphase 1. So answer is 3, A3, B5, C4, D2. This is regarding both mitosis and meiosis. Next, this is an assertion reasoning question. Bivalents are totally disjuncted from one another during anaphase 1. Bivalents are totally disjuncted from one another during anaphase 1. Now, haploid number of chromosomes move to each pole during anaphase 1. Bivalents are not completely disjuncted from one another during anaphase 1. But haploid number of chromosomes, genomes are formed, move to their each pole during anaphase 1. So A is false, R is true. Interphase. The special stage in interphase, G1 stage, S stage, G2 stage. G1 stage is known for the synthesis of RNA. So RNA polymerase is required. S stage is known for replication of DNA. So, DNA polymerase is required. G1 stage and G2 stage are known for protein synthesis. So, peptidyl transferase is required. I will repeat. Interphase is the resting phase only for the nucleus, not for the cell. It makes a cell get ready for cell division. It makes a cell get ready for cell division. It is studied in 3 substages, G1, S, G2. In G1, RNA and protein synthesis takes place. That means it requires RNA polymerase and peptidyl transferase. S stage is the most significant stage where replication of DNA occurs, so it requires DNA polymerase. Peptidyl transferase is again required in G2 phase where RNA and protein synthesis continues. So, the enzymes required for necessary for interphase will be all the 3. 1, 2, 3. RNA polymerase for RNA synthesis, DNA polymerase for replication of DNA, and peptidyl transferase for protein synthesis. Arrange the following in correct order related to meiosis: disjunction, bivalent, crossing over, splitting of centromere. Here first is bivalent because it occurs during zygote by synapses. Next is crossing over because after the formation of bivalents only crossing over takes place. Next is disjunction which occurs in anaphase 1. Finally, splitting of centromere which occurs in anaphase 2. So, the order is bivalent, crossing over 2, 3, 1, 4 because bivalent formation occurs in zygote, crossing over formation occurs in patchotin, disjunction occurs in anaphase 1. Bivalent and crossing over is prophase 1. Disjunction occurs in anaphase 1. Splitting of centromere of chromosome occurs in anaphase 2. The order is 2, 3, 1, 4. Next assertion. The division of extranuclear protoplasm is called cytokinesis. Extranuclear protoplasm is cytoplasm. So, division of cytoplasm is cytokinesis or division of extranuclear protoplasm. Extranuclear protoplasm means cytoplasm. So, extranuclear protoplasm is cytokinesis is correct. The nuclear division is called karyokinesis is correct, but both are not linked with each other. So, both A and R are correct. R is not the correct explanation of A. Answer is 2. This is another matching tabular form. List 1: August Weismann, Strasburger, Fleming, Farmer and Moore, Wirshaw. Discovered mitosis and meiosis in plants, proposed cell lineage theory, identified reduction of chromosomal number, coined the term meiosis, discovered mitosis in animal cells. Answer is 1. A3. August Weismann identified the reduction of chromosomal number in the nucleus for the first time. B1. Strasburger discovered mitosis and meiosis in plants. B1. C5. Discovered mitosis in animal cells. Walter Fleming was the scientist who discovered mitosis in animal cells. Farmer and Moore, they coined the term meiosis. 4. So, D4. They coined the term meiosis. Wirshaw is known for cell lineage theory. What is cell lineage theory? It is also called cell doctrine theory because what it means? Omnis cellulae cellula. Omnis cellulae cellula means every cell comes from pre-existing cell. Every cell comes from pre-existing cell is omnis cellulae cellula, which was proposed by Wirshaw. So, August Weismann, reduction of chromosomal number in the nucleus. Strasburger, mitosis is in meiosis in plant cells. Fleming, mitosis in animal cells. Farmer and Moore or Moore and Farmer, the term meiosis. And Wirshaw, cell lineage theory, omnis cellulae cellula. Every cell comes from pre-existing cell. Which of the following statement is true? Which of the following statement is true? Number of chromosomes in anaphase 2 is half of the number of chromosomes in anaphase 1 is wrong. Chromatids are half, not chromosomes. Number of patchotin tetrads will be double to the number of zygotin bivalents is wrong. Each chromosome shows 2 chromatids in anaphase 1 correct. Each chromosome shows 2 chromatids and 4 arms in anaphase 1 is correct. Homologous chromosomes are completely separated in diplotin is wrong. Homologous chromosomes are completely separated in anaphase 1. They are formed in zygotin and totally separated in anaphase 1. So number of chromosomes in anaphase 2 is half to the number of chromosomes in anaphase 1 is wrong. Because anaphase 1 chromosomal number is reduced to half, haploid set is formed. Anaphase 2 also shows haploid set. Number of patchitine tetrads will be double to the number of zygote in bivalents is wrong. Each chromosome has 2 chromatids in anaphase 1 is correct. Homologous chromosomes are completely separated in diplotin is wrong. They are completely separated only in anaphase 1. So for this the answer is 3. Another assertion reason. Meiosis helps in the maintenance of constant number of chromosomes from one generation to the next generation. Definitely this is correct. Why? Because meiosis is a reduction division. During the formation of zygote, gametes has to be formed. That means zygote is diploid, embryo is diploid, new plant is diploid. For the formation of zygote at one point of time, chromosomal number must be reduced to half. The chromosomal number reducing half occurs in meiosis. So meiosis helps in maintaining the constant chromosomal number from one generation to the next generation is correct. Meiosis is important in conserving the genetic integrity of the organisms. So genetic integrity of the organisms is maintained only by mitosis. Mitosis is always important in maintaining genetic integrity of the organism. So A and R are both are correct. Meiosis regarding statement is correct. Mitosis statement is correct. But because the genetic integrity is maintained in mitosis, meiosis does not help in maintaining the constant number of chromosomes. So A and R are correct. R is not the correct explanation of A. So answer is 2. Now destruction of spindle fiber during cell division results in increase in the number of nuclei is wrong. Increase in the number of cells is wrong. Increase in the number of nucleoli is wrong. Increase in the number of chromosomes is correct. This is explained as it is. When in the cell division, colchicine is used. Colchicine is a chemical mutagen used or obtained from the seeds and culms of Colchicum autumnale belonging to the family Liliaceae. Whenever colchicine is used, it does not help in the formation of spindle fibers or it causes destruction of spindle fibers. Chromosomes does not get separated and the chromosomal number doubles and polyploids are formed. So destruction of spindle fibers during cell division results in increase in the number of chromosomes. Second answer. The movement of chromosomes is, so on this which concept the movement of chromosomes takes place ante independent of spindle fibers is wrong. Without spindle fibers coming and attaching to the centromere, without the contraction of chromosomal fibers running from pole to chromosome or centromere, without the condensation and movement of spindle fibers, chromosome cannot move. Dependent upon the association of spindle fibers with kinetochores, this is correct. Depending upon the association of spindle fibers with kinetochores only, kinetochore shows the implantations of microtubules. So spindle fibers easily attach to the kinetochores. Whenever they attach, whenever they move, they also take chromosomes with them. Due to cytoplasmic streaming movements is wrong. Cytoplasmic streaming movements does not help in movement of chromosome. ATP dependent energy consuming process is also wrong. Energy conservation, formation, everything is over by G2 stage. So dependent upon the association of spindle fibers with kinetochore is correct. The movement of chromosome is depending upon the association of spindle fibers with kinetochores. Regarding phragmoplast. Phragmoplast is a barrel-shaped structure formed in between two haploid nuclei during cytokinesis from spindle fragments. The spindle fragments present in between two haploid or daughter nuclei get aggregated to form a barrel-shaped structure called phragmoplast. Phragmoplast is made up of suberin is wrong. Phragmoplast is made up of suberin is wrong. It is oriented perpendicular to the metaphasic plant is correct. It is oriented perpendicular. Definitely it is oriented perpendicular to the metaphasic plate. Golgi vesicles release pectins into it. Whenever Golgi vesicles and vacuoles release pectins into it, then only phragmoplast converts into cell plate and cell plate becomes physical and chemical changes to form middle lamella and every cell becomes two daughter cells. Golgi vesicles release pectins into it is correct. It is composed of Golgi tubulin is wrong. It is composed of Golgi tubulin is wrong. So regarding phragmoplast, which is a barrel shaped structure formed in between two daughter nuclei during cytokinesis of cell division, what are the correct options? It is oriented perpendicular to the metaphasic plate is correct. and Golgi vesicles release pectins into it is correct. So, answer is 2 and 3, third option. This is another assertion and reasoning. Events of telophase of mitosis are reversed to those of prophase. Events of telophase of mitosis are reversed to those of prophase is correct, because in prophase chromosomes are formed from chromatin reticulum, nucleolus nuclear membrane disappears. Whereas in telophase, chromosomes that have reached their respective poles become chromatin material. That means in prophase, spiralization takes place, condensation takes place. In telophase, despiralization or decondensation takes place. Nucleus appears during prophase and disappears in telophase is wrong. Nucleus always disappears during prophase and reappears during telophase is correct. So, A is correct, R is false. So, events of telophase of mitosis are reversed to those of prophase is perfectly correct. Nucleus disappears during prophase and appears during prophase and disappears during telophase is wrong. What the statement must be here? Nucleus disappears during prophase and reappears during telophase may be wrong, correct, but here A is true, R is false. The total number of spindle apparati formed during the development of 100 microspores. 100 microspores means 100 pollen grains. 100 pollen grains. 100 pollen grains means how many pollen mother cells means 100 by 4, 25 pollen mother cells. For every cell, diploid cell, to produce 4 cells, 3 times spindle fibers are formed. Remember this concept. For every one cell, to produce 4 cells by reduction, division or meiosis, 3 times spindle fibers are formed. 1 time in metaphase 1, 2 times in metaphase 2. So, for 25 pollen mother cells, how many times spindle fibers are formed means 75 times spindle fibers are formed. So, the question is total number of spindle apparati formed during the production of 100 microspores is 75. Why? For the formation of 100 microspores or 100 pollen grains, the total number of pollen mother cells required, we have to use the concept 100 by 4, 25 pollen mother cells. One pollen mother cell to produce 4 pollen grains, it shows 3 times spindle fiber formation or 3 spindle apparati formation. For 25 pollen mother cells to produce 100 pollen grains, the total number of times or total number of spindle apparati formed is 25 into 3, that is 75. So, answer is 3. Assertion. Crossing over is responsible for the origin of new species which leads to evolution. Crossing over means at the point of chiasmata, chromatid bit breaks. After the breakage, the bit get exchanged. The breakage is done by endonuclease. After the exchange, reattachment is done by ligase. This exchange of chromatid bits is exchange of parental character. Exchange of parental characters is crossing over. Crossing over always leads to recombination. Recombinations leads to variations. Variations forms the base for evolution. So, assertion A, crossing over is responsible for the origin of new species which leads to evolution is 100% correct. Crossing over results in the recombination of genetic characters causing variations in the progeny is correct. So, A and R are both linked to each other. So, the answer is both A and R are correct. R is the correct explanation of A. R is the correct explanation of A. First option is the answer. Now, arrange the following in correct related to meiosis. Correct related to meiosis. First synapsis, then terminalization. So, answer is first synapsis, then tetrads in bivalent. Tetrads in bivalent means patchitine tetrads are formed in patchitine. Synapsis occurs in zygotine. Tetrads in bivalent formed in patchitine. Terminalization occurs in last, diakinesis, and segregation occurs in anaphase 1. I will write all the stages. Leptotine is known for accumulation of homologous chromosomes at one point, which is called bouquet stage. Zygotine is known for synapsis and formation of bivalents. Patchitine is known for crossing over, formation of chiasma and many other things. Diplotine is known for thickening, contraction and other aspects of chromosomes, which is called spiralization. Diakinesis is known for terminalization. This is all for prophase 1. Now coming to metaphase 1. Metaphase 1, all the chromosomes which are irregularly scattered in the cytoplasm are brought to the center with the help of spindle fibers. So that is called chromosomal congregation. Chromosomal congregation. Anaphase 1. Anaphase 1, chromosomes get separated. Homologous chromosomes, haploid chromosomes, genomes get separated. This is called segregation or disjunction. Segregation or disjunction. Then anaphase 2. Anaphase 2 is the stage where chromosome divides at centromere and daughter chromosomes are formed. This is the correct sequence of meiosis. So whenever a question is asked in sequence, you have to remember this. Leptotin bouquet stage, homologous chromosomes grouped at one point. Zygote synapsis and formation of bivalents. Pachytene is crossing over. Diplotin is spiralization, contraction, condensation, thickening of chromosomes. Diakinesis is terminalization. Metaphase 1 is chromosomal congregation. Anaphase 1 is segregation or disjunction. And anaphase 2, chromosome divides at the point of centromere. So for this the answer is first synapsis zygote, tetrad bivalent pachytene, terminalization diakinesis, segregation anaphase 1. First option. First option is equatorial plate has DNA and proteins. Equatorial plate has DNA and proteins, definitely, because equatorial plate or metaphasic plate is formed by centromeres. Centromeres contain DNA and proteins. Metaphasic plate is formed by the centromeres of chromosome. So because it is formed by the centromeres of chromosome, equatorial plate has DNA and proteins. So both A and R are correct. R is not the correct, R is the correct explanation of A. First is the answer. Another assertion reasoning. Crossing over phenomenon is responsible for origin of new species. Crossing over leads to variations, recombinations. Recombinations leads to variations. Variations definitely cause origin of new species, which is the first and foremost step in evolution. Activity of ligase and endonuclease are seen in pachytene. Only by the activity of ligase and endonuclease you can see the crossing over, exchange of chromatid bits. So both A and R are correct. R is the correct explanation of A. First option for this. During late prophase, the chromosomes are arranged. Late prophase. Early prophase is known for the conversion of chromatin into chromosomes. Chromosomes containing one centromere, one chromatid, two arms. During mid prophase, the chromatid arm of each chromosome splits longitudinally so that every chromosome gets one centromere, two chromatid and four arms. During late prophase, the chromosomes come to the corners, nucleolus, nuclear membrane disappears, and chromosomes are randomly distributed in cytoplasm. So regularly one above the other is wrong. Regularly on a single horizontal line is wrong. In a line in a descending size order is wrong. Randomly distributed in cytoplasm is correct. So answer for this is 4. The subphases of prophase 1 are respectively, this is the expected sometimes in MSET. Subphases are correct sequence is leptotin, zygotin, pachytene, diplotin, diakinesis. The answer is given as 3. It is not 3, it is 4. Leptotin, zygotin, pachytene, diplotin, diakinesis. So fourth is the option, not 3. Fourth is the option. Repulsion between homologous chromosomes is started in one of the following stages of meiosis. Repulsion between homologous chromosomes. Repulsion between homologous chromosomes always occurs in diplotin. Why? Because in this stage only an enzyme called synaptinymal complex starts disappearing or disappears. Starts disappearing or completely disappears. So, in this stage of meiosis, particularly diplotin, synaptinymal complex starts disappearing. Whenever synaptinymal complex starts disappearing, definitely there is a repulsion between homologous chromosomes. So, pairing between homologous chromosomes takes place in zygotin. Crossing over between homologous chromosomes occurs in patchotin. Repulsion between homologous chromosomes starts in diplotin. Leptotin is absolutely no homologous chromosome pairing. Diakinesis, terminalization, anaphase 1, disjunction between homologous chromosomes. Diplotin is the repulsion between homologous chromosomes. Why? Because in this stage only the dissolving capacity of our synaptinymal complex enzyme completely disappears. Chromosomal spindle fibers are attached to centromeres during. In prophase, chromosomal spindle fibers are not formed. In telophase, chromosomal spindle fibers presence is totally absent. So, in 3 options, prophase, telophase are given. That means 1, 3, 4. Metaphase and anaphase. In metaphase, importance is special formation of spindle fibers and orientation of chromosomes at the center. So, metaphase definitely chromosomal spindle fibers are formed. In anaphase, by the contraction of chromosomal spindle fibers only, the daughter chromosomes are formed and they are moved to their respective poles. So, metaphase and anaphase are the stages where chromosomal spindle fibers are attached to centromeres. The total number of mitotic divisions that occur in the formation of 32 cells from a single cell. This type of mitosis problems, we have to use the formula n minus 1. So, 32 minus 1, 31. So, every time you have to use this formula. Why? Because mitosis means first cell gone and remaining cells are formed. So, suppose the same question is asked, the total number of mitotic divisions that occur for the formation of 256 cells at the root tip, 256 minus 1. So, here the answer is the total number of mitotic divisions that occur from the formation of 32 cells from a single cell is 31. The formula used in this is n minus 1. The following minimum number of meiotic divisions has to occur for the formation of 100 paddy grains. 100 paddy grains. 100 paddy grains means paddy grain is a caryopsis, dry indication fruit. Caryopsis is a single seeded fruit. Single seeded fruit. For this there is a formula n by 4 plus n. So, for the formation of 100 seeds, 100 by 4 plus 100. So, meiotic divisions required is 125. This formula is very important, n by 4 plus n. Any time when seeds are formed, 100 seeds means 100 ovules are required. 100 ovules means each ovule show 1 meiotic division, so 100 meiotic division. 100 say ovules means 100 pollen grains. 100 pollen grains means 100 by 4, 25. 100 plus 25, 125. That much long calculation is not that better. N by 4 plus n, 100 seeds by 4 plus 100. So, answer, how many meiotic divisions are required to produce 100 paddy grains? Here 100 paddy grains, we have to think about Cypsel caryopsis fruit. Caryopsis fruit is single seeded. So, for the formation of seeds, for 100 seeds, how many meiotic divisions are required can be asked. Just to confuse, 100 paddy grains is given. So, 100 by 4 plus 100, 125 is the answer. If it is given as following minimum number of meiotic divisions to produce 100 seeds, we will do it usually by using n by 4 plus n. But to confuse the people, it is given as 100 paddy grains. 100 paddy grains means 100 caryopsis fruit. 100 caryopsis fruit is single seeded. So, single seeded fruit means 100 seeds. 100 seeds means 100 by 4 plus 100, that is 125. It is a 3-column matching. Stage of cell cycle, period of hours, main change. G1 stage 5 hours, increase in the number of cell organelles. Increase in the number of cell organelles does not occur in G1 stage. S stage 7 hours duplication of genetic material. Duplication of genetic material, that is DNA. G2 stage 3 hours synthesis of energy pool is correct. M stage 1 hour division of centromere is correct. So 3 and 4, not duplication of genetic material, it is duplication of only DNA. So answer is 3 and 4, that is the fourth option. Coming to a matching. Prophase, metaphase, anaphase, cytokinesis. Cell plate formation, division of centromere, vanishing of nucleolus, equatorial plate formation, entry of nuclei into interphase. A3, prophase, vanishing of nucleolus means disappearance of nucleolus. Metaphase, B4, equatorial plate formation. Anaphase, C2, division of centromere, formation of daughter chromosomes. Cytokinesis, cell plate formation. Phragmoplast is formed. Vesicles and vacuoles of Golgi complex accumulate in phragmoplast, secrete pectins into it, converts into cell plate. Cell plate by physical and chemical changes become middle lamella. So answer is prophase, vanishing of nucleolus. Metaphase, equatorial plate formation. Anaphase, division of centromere. And cytokinesis, cell plate formation. First option. Arrange the following events of meiosis in reverse sequence: daughter chromosome formation, synapsis, disjunction and endonuclease. Reverse means from behind. Daughter chromosomal formation, anaphase 2, first option. 3. Disjunction, anaphase 1. Endonuclease action, patchtin, synapsis, zygotin. So reverse action we have to write it from behind, from anaphase 2. So daughter chromosome formation occurs in a reverse fashion first, then disjunction occurs, then endonuclease action occurs, then synapsis occurs, and anaphase 2, anaphase 1, patchtin, zygotin. So answer is 1, 3, 4, 2. Arrange the following sequence of prophase 1 in correct sequence. First is here leptotin, zygotin, patchtin, diplotin, diakinesis. Here first is zygotin, then patchtin, then diplotin, diakinesis. So 4, 2, 3, 1. 4, 2, 1, 3. Sorry, 4, 2, 1, 3. First option. Zygotin, patchtin and other things. Next. Genomes of meiocytes are separated during. Genomes of meiocyte ante diploid cell. Genomes get separated. If you see the diagram like this, the haploid set of chromosomes with 1 centromere, 2 chromatids and 4 arms. 1 centromere, 2 chromatids and 4 arms, like this. Here this is a genome and this is a genome. These genomes are pulled towards their respective poles by the pulling action of spindle fibers in anaphase 1. So anaphase 1, every time every chromosome contain 1 centromere, 2 chromatids and 4 arms. So these are called genomes, and these genomes are pulled towards their respective poles by pulling action of spindle fibers, which is popularly known as disjunction or segregation. This occurs during anaphase 1. So genomes of a meiocyte are separated during which stage means anaphase 1. Zygotin is known for. Chromosomal condensation is initiated is wrong. Chiasma formation is wrong. Chromosomes get divided is wrong. Chromosomal condensation initiation in prophase. Chiasmata formation occurs in patchtin. Chromosomes get divided in anaphase 2. Spindle fiber formation is correct. Zygotin is known for spindle fiber formation and enlargement of nucleolus. Next. Reciprocal changes of mitosis takes place in one of the following sets. Reciprocal changes means the changes that occur opposite to each other in prophase and telophase. In prophase, chromatin reticulum transforms into chromosomes. In telophase, chromosomes converted into chromatin reticulum. In prophase, nucleolus and nuclear membrane disappear. In telophase, nucleolus and nuclear membrane reappear. So the correct exact opposite changes occurs in prophase and telophase. Homologous chromosomes are What are homologous chromosomes? They are obtained from parental generations. Morphologically and genetically similar, wrong. Same parent is wrong. Morphologically, genetically different chromosomes obtained from same parent is wrong. Morphologically and genetically similar chromosomes obtained from different parents is correct. Different parents is correct. Different parents, chromosomes obtained from different cause. Morphologically and genetically similar chromosomes obtained from different parents are popularly called homologous chromosomes. 3 is the option. Chromosomes divides in which stage of meiosis? Meiosis is known for 2 divisions in nucleus, 2 divisions in nucleus, 1 division only in chromosomes. 2 divisions in nucleus and 1 division only in chromosomes. This 1 division in chromosomes occurs during anaphase 2. So, anaphase 2 is known for the division of chromosome at the point of centromere. So, in meiosis, where do the chromosomal division occurs? In anaphase 2, chromosome does not divide at the point of centromere and no question of formation of daughter chromosome. Chromosomes divides only at the point of centromere during anaphase 2. Next. DNA duplication during which stage of cell cycle? Cell cycle is known for interphase and mitotic phase. Interphase is known for G1 stage, S stage, G2 stage. G1 stage is known for RNA and protein synthesis. G2 stage is known for RNA, protein synthesis, doubling of cell organelle, and also energy pool is created and stored for the future use of formation of spindle fibers and movement of chromosome. S stage is the most significant stage where DNA duplication takes place. So, the answer for DNA duplication takes place during the following cell cycle is S stage. It is an event of telophase. Formation of daughter chromosomes is wrong. Formation of daughter chromosome is an event that occurs in anaphase. Division of centromere, anaphase. Formation of spindle fibers is metaphase. What is the event of telophase? Formation of daughter nuclei. Formation of daughter nuclei always occurs in telophase. The phenomenon of meiosis that helps in the origin of new species and leads to evolution. The phenomenon of meiosis helps for origin of new species and leads to evolution is crossing over. Synapsis, pairing of homologous chromosomes, formation of bivalents. Disjunction, movement of homologous maternal, homologous paternal chromosomes converted into genomes and to various poles. Disjunction or segregation, both are same. So, the phenomenon of meiosis helps for the origin of new species and leads to evolution is crossing over, which occurs in patch 10. Crossing over leads to recombinations, recombination leads to variations, variations leads origin of new species, and that helps in evolution. No 2 persons look alike is origin of new species by crossing over. So, these are some of the questions in meiosis. These are some of the aspects regarding nucleus, chromosomes, nucleic acids, mitosis, meiosis. This is about cell division, and we discuss the next chapter, tissues, anatomy and other things in the next class. So, I wish you all the success for the children.